So good afternoon, everyone. So today my presentation is going to be under the title of Spatiotemporal Transcriptome Profiling Unveils Multi-Scale Heterogeneity Levels Between Subpopulations of Bacillus subtilis surface-associated communities. So my presentation is going to be as follows. I'm going to start by a brief introduction, followed by the objectives, and then a series of workflows with some results, and finally a small conclusion. So to start, surface-associated multicellular assemblages, known as biofilms, are recognized as the predominant bacterial lifestyle in nature, where Bacillus subtilis is a gram-positive multi-spore-forming bacterium, which is frequently found in the soil, digestive tract, and has been exploited by several man-made applications, such as fermentation of food, restoration of historical buildings, commercially used as a biocontrol agent, and also as a bioprotective agent for positive surface colonization. Now, on the other flip, this surface colonization by non-pathogenic bacteria could be highly resistant to disinfectants used, and thereby could increase the possibility to act as a protective shield for pathogenic bacteria, a scenario that could be present in industries, medical sectors, and also it could reach to space stations. Now, what is a biofilm? A biofilm is a complex system where composed of cell aggregates that are embedded in a self-produced matrix that are adherent to each other and/or to a surface. Now, by such an aggregation, cells residing in a biofilm could acquire new emergent properties that were not present or not predictable by studies on free living bacterial cells, such as we have resource capture by sorption, enzyme retention, social interaction, tolerance and resistance, and also localized gradients due to environmental microscale adaptations, which will lead to cell physiological heterogeneity level hindered behind a sophisticated network of gene regulation. So in the laboratory, several artificial conditions have been used to study Bacillus subtilis biofilm formation, including the colony at the air-liquid interface. And when the agar surface is a semi-solid one, in this case, motile bacterial cells can swarm over the surface by an organized collective movement while proliferating and consuming nutrients. We also have the floating pellicle, the floating biofilm known as pellicle at the air-liquid interface. And lately, when Bacillus subtilis has been found to adhere to abiotic surfaces, this has grabbed the attention for the submerged biofilm model. Now, in this context, an architectural competitive submerged biofilm study has been done on different Bacillus subtilis strains, including the 168 reference strain, where Bacillus subtilis NDMed has been shown to form the highest biovolume on the submerged level. Now, this strain, the Bacillus subtilis NDMed, is a strain isolated from an endoscope washing disinfector from a hospital in England. And by the use of scanning and electron microscopy techniques, it has been found that this strain forms compact and thick biofilms, as we can see here from the shadow, which represents a cross-section through the XZ plane. And it also produces high amount of exopolymeric substances when compared to the 168 reference strain. Now, the Bacillus subtilis by itself, it is not a pathogenic bacteria, but it has been found to protect pathogenic bacteria like Staphylococcus aureus in mixed biofilms and to be hyper-resistant to the action of biocides. Hence, understanding how these surface-bounded communities are formed is crucial for the development of suitable strategies for their treatment and for their control. So the aim of this project was to compare the different biofilm models of Bacillus subtilis and to identify genetic determinants involved in the subpopulation heterogeneity. And this has been done by three parts. So the first part was to select the different localized heterogeneous compartments, and then a spatial transcriptome analysis was done for these samples or for these compartments, and then an in situ spatiotemporal monitoring was also done. So I will start by the selection. So for this selection, we have selected to study a 24-hour static liquid culture, where we have separated the pellicle from the detached cells from the submerged one. Moreover, we have used a 24-hour swarming plate from which we have separated 4 localized compartments. The mother colony, which resembles a mature macrocolony on agar, we have the base of the dendrite, which resembles an early biofilm form. And we have the dendrites here that are a monolayer of cells that are getting ready to start producing the matrix to form later a biofilm. And we have the tips that are the highly motile and highly dividing cells. And also as a reference, we have collected from the planktonic culture the exponential and the stationary phase. So now in order to go further, to understand the core of the transcriptional network that takes place during biofilm development, a spatial level gene expression was done. So after this selection, so we finally had 9 different conditions. With the 3 biological replicates, it is important to note here that all the cultures were performed in the same minimal glucose B medium. So for the 27 samples we have here, RNA extraction by mechanical lysis was performed. And then it was an assessment of the quantity and the quality of the RNA by nanodrop and bioanalyzer was also done, to be sent afterwards to sequencing in the UWC platform. So now, after sequencing, we have received a huge RNA data set. So in order to assess the quality and the reproducibility of this RNA data set, a hierarchical clustering analysis was performed. So this clustering here is formed according to some notion of similarity, from which we can see here that the mother colony, for example, and the stationary phase, they are not only distinct from each other, but also from all other compartments. It could also show that the exponential phase is closer to the swarmers, and then the three adjacent compartments in the 24 static liquid culture were also clustered together. Now, this clustering also can show us how the 3 biological replicates that we have are clustered together, are very close to each other. This is true for all, except for the adjacent compartments that we have for the swarming. This could be for 2 reasons. Either technically, it was difficult to differentiate between these compartments, or the physiology of the cells of these compartments is very close. So in order to identify the physiology of the cells, so we have performed a differential expression gene analysis, which show us the number of the genes that are statistically upregulated by a log2 fold gene. Here it is in the yellow. And those that are statistically downregulated in blue in a compartment compared to the other taken as a reference. So when we have a look, for example, on the compartments here, the base, the dendrites, and the tips, where they were clustered here, we can see that there is still some genes that are statistically significant between these compartments. Now here is the heat map for the global, to have a global view of all the transcriptome that we have. So this heat map governs all the genes. We have in the Bacillus subtilis and the MED 4,200 genes. So the clustering here is according to the similarity in the expression of the genes. And as mentioned, the yellow is for the upregulation, the blue is for the downregulation, taken as a log2 fold chain, calculated according to the average of all the compartments except the stationary phase. So we can see by this heat map that we have some blocks of up and down regulation. If we look at this cluster, for example, this cluster governs 80% of the genes that are related for sporulation, where we can see that it is mainly in the pellicle and in the mother colony. This group here, it requires for protein synthesis and modification and degradation, which is mainly upregulated in the exponential phase, and in the swarmers, the ones that are actively dividing. This group here is for motility and chemotaxis, so we can see also it is upregulated in the planktonic, and also during the swarm, and also in the detached cells, while it is downregulated in the 3 biofilm models that we have. This here group is also for the, it governs 40% of genes related for biofilm formation. Now, while we are digging into the transcriptome data and looking for the different pathways, the regulatory pathways, so the carbon metabolism pathway has grabbed our attention. Knowing that glycolysis and gluconeogenesis are 2 oppositely regulated pathways, where in Bacillus subtilis, growing in glycolytic conditions, such as we have with the glucose. So we will have the glycolytic process will be on, and for instance, the CGGR GAP-A operon will be induced, and this gene will be repressed in the presence of gluconeogenic conditions. Now, in the purely gluconeogenic conditions, we will have, for example, the PCKA and the GAP-B will be de-repressed. So thus, GAP-A and GAP-B are two oppositely regulated genes that cannot be expressed together in the same cell. So if we look at these two groups, the GAP-A and the CGGR, and here we have the GAP-B and the PCKA, we can see that they are oppositely regulated in all conditions except in the submerged model. So this can suggest that in the same biofilm, different subpopulations are entering into different metabolic pathways. So the question that arises here is that how are these subpopulations localized, or how are these formed in the biofilm? Are they stratified, segregated, or intertwined? So in order to go beyond to the local gene expression to monitor in situ the single cell gene expression inside a biofilm community, so the strategy that we have depended is that from the transcriptome data, we have searched for genes that could play as a good candidates to answer our biological question, such as those genes related for the carbon metabolism, the matrix. We also took some motility and so on. We have reported these genes transcriptionally either by GFP or M-Sherry. And in some cases, when we had these oppositely regulated genes, we were able to report them differently in the same cell to observe under the confocal microscopy. So here, this transcriptome data here, it is the same as the previous one, but it's just a quantitative data in a log 2-fold chain. This is the CGGR and this is the GAP-B. So we can see that they are all oppositely regulated, except in the submerged, both of them are upregulated. So, in a single NDMET strain, we were able to report the CGGR by M-Sherry, CGGR, which is required for glycolytic and glycolytic pathway, and the GAP-B in GFP, which is for the gluconeogenic, and we have observed them in situ after 24 hours. So here we can see that these images really reflect what we were able to see in the transcriptome data. So for example, we can see here in the mother colony where the glucose starts to be limited, we will have the intensity of the glycolytic gene, which is reported by M-Sherry. It becomes less than that during the swarm, while we have an upregulation, or we have an expression of the gluconeogenic gene, the GAP-B. To have a zoom on the mother colony, for example, we can see how these two subpopulations are spatially localized, where the yellow reflects the superposition of the red and the blue and the green. Sorry. Now, to have a look to make a zoom on the base, for example, here we can see how the conversion from the glycolytic to the gluconeogenic condition starts by a single cell that starts to produce the gluconeogenic gene in a population of a glycolytic expressing gene. So here it is the same. We had a look on the liquid culture after a 24-hour liquid culture, where also we can see that in the pellicle, we have most of the population is expressing glycolytic, with some cells that are expressing brutally this gluconeogenic gene. And here we can see for the submerged, both of them are upregulated, seems to be upregulated. Now, to have a further or a deeper look to the subpopulation, we have monitored the subpopulation for 3 days. So at the beginning, we see that because we are working in glycolytic condition, so the cells are going to express the glycolytic gene, and then it increases, and at a certain level it decreases, where the glucose starts to be limited, and then we can see we have an upregulation in the gluconeogenic related genes. So, and we noticed that also after prolonged incubation, these two subpopulations will still coexist together. Now, another example for this construction of the two oppositely regulated genes, also here in the NDMed, it has reported by GFP on the promoter of HAG, which is required for motility, and M-Sherry on the promoter of TAP-A that is required for the matrix production. So in this movie also, we can see that at the beginning, we have a building of sessile chain cells with the production of matrix. And then after several hours, here in this case, it was like around 5 hours, we have an explosion or de-differentiation from the sessile cells to motile cells. And these motile cells are going to form the pellicle afterwards at the air-liquid interface. So to see it clearly, because it could be very fast, here it's an illustration of a chemograph. This chemograph illustrates how it shows the intensity of the gene as a function of time and space. So we can see for the HAG, for the motility, at the beginning it was not really highly expressed. And after 5 hours here, we have like an explosion of these fragmented motile cells. And then we have, another wave here that seems to be expressed in a mature biofilm afterwards. And here for the TAP-A, we can see that it was highly expressed at the beginning, and then it was decreased, and then only increased a little bit, expressed a little bit afterwards in a very mature biofilm. So since we are interested in the biofilm, so we have also reported further other matrix-related genes. So here it is just as a reference, we have used NDMed, which was constitutively reported by GFP. And here are the different matrix-related genes. So this shows that the matrix expression is not a uniform one, but as a dynamic mosaic with a different spatiotemporal patterns of heterogeneous gene expression, where, for example, we have the EPS and the TAP-A are mainly expressed at the beginning. The BSLA and the SRF, for example, seems to be an intermediate, like in a transient phase, and the YPQP and the CAP, for example, they are a late matrix produced genes. So to sum it up, to sum everything up, so the RNA sequencing is a powerful tool for the fact that the twin aspect of the discovery, from which we can detect and select some genes of interest, and also for the quantification, which usually give us an idea about the average, or an idea about the average of this population that we are studying in a single high throughput sequencing assay. However, to go from the transcriptome level to the in situ functional cell level, in our case, there are several techniques, but in our case, we have selected to do the transcriptional fusions combined with the confocal microscopy techniques. However, there's also another technique known as, it is a very recent, it is the PAR-seq fish technique, which is used to capture gene expression profiles at a high throughput level with preservation of the physical context of the individual cells within a biofilm. So all this work has been done in the laboratory of Romain Briondé, and Michaelis here in Juillan-Josas. So I would like to thank all who have I worked with, all the contributors, and also for the platforms, for the I2BC, for the sequencing, for MIMA2, for the confocal imaging, and also for all the funding for this work from the Union Southern Suburbs of Municipality of Beirut. We also have the INRAE, Campus France, and AgroParisTech. And I would like to thank you all for giving me your attention. And I would like to hear your questions.